Okay, this is slide number 58 of the chapter 8 slides, and we are going to discuss ATP, which is adenosine triphosphate. It is composed of a ribose, which is a 5-carbon sugar that you find in the nucleic acid RNA. You know, there's RNA and there's DNA. RNA has the sugar ribose, whereas DNA has deoxyribose. So ATP has a ribose sugar, like RNA. It has adenine, which is a base, and then 3 phosphate groups attached. So it's similar to adenine of an RNA molecule. All right, and the important thing about ATP is that it has those 3 phosphate groups attached. So here's your adenine, which is a double ring structure. Here's your ribose sugar, and then instead of just 1 phosphate group, there are 3 phosphate groups attached in adenosine triphosphate. There is an animation, which is slide number 60, if you want to watch it, and also another one on slide 61 and 62. That they are animations regarding ATP. Now, the way that ATP releases energy is when the 3rd phosphate or the terminal phosphate bond is broken by hydrolysis or breakdown by the addition of water. So basically, when a phosphate is released from ATP, then energy is released. That's how a cell would get energy from ATP, is when that 3rd phosphate is released. So basically, when ATP has water added to it, and the water is used to remove that 3rd phosphate. So you would end up with an inorganic phosphate plus ADP, adenosine diphosphate, plus the energy that's released, because this is an exergonic reaction. And so this is what happens to release energy from ATP. And cellular work in the form of mechanical work, transport work, and chemical work is powered by ATP breakdown or ATP hydrolysis. So the energy, and this is important because this is definitely more than one question in your homework assignment, that you need to know that the energy from the exergonic reaction, the exergonic breakdown of ATP, is used to drive endergonic reactions, or reactions which build larger molecules, anabolic reactions. So that's what is meant by coupled reactions, okay? So exergonic reactions are coupled to endergonic reactions by ATP, basically, is what you need to know. Phosphorylation is the transfer of a phosphate group from ATP to another molecule, and that is typically used to power endergonic reactions. So you definitely don't have to do any math with change in G, that's for sure, so don't worry about that. Okay. All right, you can watch this video on ATP metabolism, on metabolism, an overview of metabolism. That is slide 73. And then we're going to move on to enzymes. We're going to talk about enzymes. So you will have, just to summarize what you've learned, what we've covered so far, you will have questions on entropy, but they will just be general questions. There won't be any calculation questions, and you won't have to, there won't be any questions on change in G. In other words, whether or not a reaction is spontaneous, that kind of thing. That's a little bit more chemistry than you need to know for biology. So you just need to know that entropy decreases when the reaction is an anabolic reaction or endergonic reaction. So entropy decreases during anabolic and endergonic reactions, and entropy increases if the reaction is catabolic and exergonic. You might want to replay that, but that's important to know, and that's really all you need to know about entropy or disorder, okay? Now we're going to move on and talk about enzymes. So a catalyst is a chemical that speeds up a chemical reaction without being consumed, and an enzyme is a macromolecule, almost always a protein, that is a catalyst to a specific reaction. The enzymes are typically named for what they do. So sucrose is a sugar molecule. It's a disaccharide. The enzyme which breaks down sucrose is called sucrase. And this is how it works. Here's sucrose, which is called your substrate. In the presence of water and your enzyme sucrase, the sucrose will bind to the active site here on sucrase, and then that will cause the breakdown of sucrose into glucose and fructose. So the activation energy, you will need to know the definition of this term. The activation energy is the energy needed to break the bonds of the reactants. It's called the activation energy. And the way enzymes work is they lower this activation energy. And when you lower the activation energy, the reaction occurs faster. Okay? So, So if you look at an exergonic reaction, you have your reactants here on the left, and they have a certain amount of energy, and their activation energy is the energy needed to break the bonds between the atoms in those reactants. So there's a certain amount of activation energy that's needed to cause those bonds to break so they can form into products. So the reactants have more energy than the products, but there's still activation energy needed to cause their bonds to break. So an enzyme would lower that activation energy. So an enzyme catalyzes a reaction or speeds up a reaction by lowering the activation energy barrier enough for the reaction to occur at moderate temperatures. And this shows you the effect of the enzyme. The reactants in the products have the same amount of energy, but the activation energy, the energy needed for the bonds to break in those reactants, is lowered by an enzyme. So enzymes lower the activation energy, and that's going to cause the reaction to happen faster. There's an animation on how enzymes work in slide 85. And here's some definitions you'll need to know. The substrate is the reactant. Okay, so anytime, if we go back to the reaction with sucrose, sucrose was the substrate, and then the products were glucose and fructose. Most enzyme names end in ase, A-S-E, like sucrase. And the active site is the region on the enzyme where the substrate binds, where the reactant binds. What happens when the substrate binds to the active site is that causes the enzyme to change shape just slightly, and it tightens around the substrate. You can think of it like a handshake. This is called the induced fit. The induced fit results from the interactions between the chemical groups on the substrate and the active site. And that's what causes the reaction to speed up. You can see a picture here. Here's your active site, which is kind of large here before the substrate binds to it. Once the substrate binds to it, you have the enzyme substrate complex that forms, and so the enzyme tightens around that substrate. All right, here is the cycle of how enzymes work. Basically, step 1, the substrate enters the active site. Here are your substrates, the red and green molecules. They enter the active site. Step 2, they are held in the active site by weak interactions. So there's kind of a small shape change where the enzyme kind of hugs the substrates a little tighter. In step 3, the activation energy is lowered by the enzyme. The substrates are converted to products. As you can see, they're different colors, so they're different chemicals. Those products are released, and then the enzyme can perform that reaction again. The enzyme is not changed by the reaction. Here's another animation on enzymes, slide number 93. So how can we lower activation energy? Substrates may be oriented to facilitate the reaction. Substrates may be stretched to make bonds easier to break. The active site may provide a microenvironment that favors the reaction, or amino acids in the active site may participate in the reaction. So these are 4 different ways that enzymes lower activation energy. Mainly, you just need to know that they do lower activation energy. That's how they work, is they lower the activation energy for a reaction. Now, you can speed up a reaction by increasing the amount of substrate, increasing the concentration of substrate. In other words, add more reactant or substrate, you're going to get more product. And whenever we have increased the amount of substrate, and the enzymes that are available to react with the substrate are all bound to substrate, we say that their active sites are all engaged. We say that the enzyme is saturated. So if the enzyme is saturated with substrate, adding more substrate will not increase the rate of the reaction, but adding more enzyme will. Now, this is one of your homework questions. So if your enzyme is saturated with substrate, you can't increase the rate of the reaction by adding more substrate, but you can speed up the reaction by adding more enzyme. You can also alter the temperature and the pH. Typically, when you raise the temperature, that'll speed up a reaction. Every enzyme has an optimal temperature, like optimal means best. So every enzyme has a temperature at which it works best. So if you raise the temperature of the reaction to that optimal temperature, not higher than that, but to the optimal temperature, that can raise the rate of the reaction. But if you raise it higher than the optimal temperature, the enzyme can denature, and then it won't work at all. The reaction won't occur because the enzyme will change shape and it won't be able to do its job, because its role depends on its shape. Its function depends on its shape. This is just showing you the 2 different optimal temperatures, one for a human enzyme, which is around body temperature, and then one for a heat-loving bacteria, a thermophilic bacteria. Every reaction has an optimal, or every enzyme has an optimal pH. The optimal pH for most enzymes in the human body is around 7.4. Which is about the same pH as our blood. But in the stomach, there is an enzyme called pepsin. It's a stomach enzyme. And in the stomach, the optimal pH for that enzyme pepsin, which begins the breakdown of protein, the optimal pH is 2. And stomach acid helps to decrease that pH. So here you have 2 different enzymes, pepsin and trypsin. Pepsin is found in the stomach, and its optimal pH is 2. So when the stomach pH drops down to 2, when there's enough acid in the stomach, so the pH is lowered all the way to 2, which is very, very acidic, very low, then pepsin is activated. Trypsin is an enzyme in the intestine, and that one, it has the optimal pH of 8, which is slightly basic. All right, you do need to know what cofactors and coenzymes are. Cofactors are non-protein helpers that bind to the enzyme permanently or reversibly with the substrate. They can include zinc, iron, copper. They can include metal atoms. And then if they're organic cofactors, they're called coenzymes. And vitamins can act as coenzymes. So they just basically increase the enzyme's activity, or increase its, make it more effective. Inhibitors are going to inhibit enzymes. So if they are competitive inhibitors, they closely resemble the substrate, and they can actually bind to the enzyme's active site, and that will inhibit the activity of the substrate forming into product, because it's preventing substrate from binding to the active site. So if it's a competitive inhibitor, it resembles the substrate and it can bind to the enzyme's active site. See, the competitive inhibitor basically blocks the substrate from binding to the active site. And there is an video animation on slide 108 on competitive inhibition. Now, if it's a non-competitive inhibitor, that means it's still an inhibitor. It still inhibits the enzyme, but it does this by binding to another part of the enzyme, not the active site, but away from the active site. And it'll cause a shape change in the enzyme, so it will make it less effective. And here is shows you non-competitive inhibitor. Doesn't bind to the active site, but it binds somewhere else, and the enzyme changes shape, and so it doesn't work as well. And there's an animation on non-competitive inhibition on slide 111. Here's an example. Toxins and poisons are often irreversible enzyme inhibitors. For example, sarin gas was used in a chemical attack in Syria in 2017, killing and injuring hundreds. Sarin binds covalently to the active site of acetylcholinesterase. That's an enzyme that is important in the nervous system. It affects, it breaks down ACH, acetylcholine, which is a neurotransmitter in the nervous system. So in other words, it stimulates nerve impulses. So by binding to the active site, the enzyme cannot break down acetylcholine, and it causes the nerves basically to stay, to keep firing, the nerve impulses to keep firing. So they don't really, the nervous system doesn't really slow down. And that is bad. That's a bad thing. So we want nerve impulses to happen when they're needed, but we don't want them to keep on happening. We don't want continuous stimulation, I guess you could say. All right, enzymes are proteins. So proteins are coded for by genes. Genes code for specific proteins. That's what they do. A gene is a piece of DNA that codes for a specific protein. So changes in genes or mutations will lead to changes in the amino acid of the composition of the enzyme. So if even one of the amino acids changes, it can change the shape of the enzyme and change how it functions. So it can be beneficial or it can be harmful, because the change in shape can be a good thing or a bad thing. But here's an example. It says repeated mutation and selection on the beta-galactosidase enzyme in E. coli bacteria resulted in a change of sugar substrate under lab conditions. Okay, then just very quickly, we're almost finished, so I'm just going to keep going. Allosteric regulation is when you have a regulatory molecule that binds to a protein at one site and affects its function at another site. So that can cause inhibition or it can cause stimulation of an enzyme. So allosteric is when a molecule binds to a protein at one site and affects the function at another site. And I'll show you a picture of how that works in case you have a question about it. So an allosteric enzyme with 4 subunits. Okay, here's the enzyme, and it's got 4 different active sites. Okay, so 4 different substrates can bind to that enzyme. Now, an activator can bind to the enzyme and cause it to be more effective. See how it binds here? The activator binds here, which is not at the active sites. It's like a different place, but it actually causes that enzyme to be more effective and more likely to bind to its substrate. Then you can have an inhibitor, which binds to the enzyme and changes its shape so that it's not going to be the right shape to bind its substrate. So that's basically these allosteric regulators can inhibit or they can activate. Here's an activator that causes the enzyme to work better, and here's an inhibitor that causes the enzyme not to be able to work, not to work anymore. And then there is this concept of cooperativity, which is when the substrate binds to one active site, triggers a shape change in the enzyme that stabilizes the active form for all other sites. And this amplifies the response of the enzyme. So let's see if there's a good picture. All right, so cooperativity, you have the inactive form of the enzyme. When the substrate binds to the active site, it now converts it to its stabilized active form. So this binding of the substrate is causing the enzyme to be more effective. It's like allosteric activation, but it's caused by the substrate itself binding to the active site. And that's cooperativity. And then there's feedback inhibition, which is when enough product is produced, it will inhibit the enzyme so that it won't keep producing product. That's feedback inhibition. All right, and so we are going to stop here. And this is for the Chapter 8 slides.